Hello, guys, and welcome to our next installment of this week's lecture. Hopefully that test went okay for you guys, and we're going to talk about the immune system. I'm going to share my screen starting now. Hopefully it's going to work. Let me make sure. Okay, cool. No, it's not sharing. All right, hold on. One of these days I'll figure it out. Okay, it's not letting me figure it out the way I want to, so we're going to do it like we did the last time. Okay, so we're going to talk about the immune system, and the immune system comes in two different sets. We have what we're born with, innate, and adaptive, which is what our body learns from adapting, okay? So our innate defenses are our surface barriers. That includes the skin. Oh, yes. Puts the lotion in the basket. It does what it's told. Or mucous membranes, so mouth, and other places that I'm not going to show you where mucous membranes are. And then we have our internal defenses that we are born with. So that's the phagocytes. Those are the macrophages that we learned about. They go chomp, 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 like not Pokemon, Pac-Man. We have natural killer cells. They're exactly what their name says. They naturally kill things. We have inflammation. So if our body has a foreign invader, our body does things to make a signal like, woo, woo, red alert, something's not there. There are certain proteins that our body makes that are antimicrobial, and then our body has a fever. Our body's naturally born ability to have a fever to get rid of things. Then we have adaptive. So there's two types of ways that our body has learned that we learn and adapt to immune. We have humoral immunity, which is the basis of most vaccines, which is through B cells or antibodies are being made. And then we have cellular immunity, which is using your T cells. And that we'll get into in a few moments. So first line defense, we got our skin. The skin that we're in will protect us within. Anywho, so why is the skin such a great barrier? So we have acid of the skin, believe it or not, that inhibits microbe growth. We have certain enzymes. So even not just skin, but the mucous membranes. We have respiratory tract. We have a bunch of enzymes in there that kill bacteria and pathogens. We have lysozymes in our saliva. So we're learning from dental anatomy, and their job is to kill things that we eat and bring into our mouth or breathe in. Our tears have lacrimal enzymes that kill microorganisms so we don't get pink eye. Our stomach has enzymes so we don't get microorganisms there. Then we have mucin, which is mainly found in the digestive and respiratory tracts and helps trap things. We have something called defensins, which are proteins or parts of proteins, peptides that help inhibit any kind of microbial growth. And then the oil that our skin produces actually has a purpose, not to just give us acne and yucky skin. Its a job is that the lipids in the skin and something called dermacidin in our sweat, they're meant to be toxic to some bacteria. So everything has a purpose, okay? And then our second line defense, we have a bunch of chemicals and cells that go do as well. Okay, so we have those phagocytes, like I said. So phagocytes are, we don't need to learn how to make them. Our body is naturally born with them. Our phagocytes go in. They sense that something is foreign and invading and go chomp, chomp, chomp. We have natural killer cells. Their job is just to be like suicide bombs. We don't like you, boom, you're out. Inflammatory responses. So we have macrophages, which are also a type of phagocyte. We have mast cells that are responsible for allergies, white blood cells, and then we have a few chemicals we'll talk about. We have proteins called interferons and complement proteins that help our immune system out. We have a fever. Fever is our body's natural response. Don't fight a fever. Your body's doing what it's supposed to do. And if our body, how does our body know what's foreign, what's not? There's this thing called MHC component. And MHC is a special type of protein that's expressed on human cells. Guess what? Non-human cells don't have it. And that's how our body can recognize self from foreign. Okay? And then on our cells as well, not only do we have this little MHC, which tells us that we're human, we also have something called toll-like receptors. And what they do is they help identify what's foreign and triggers the specific immune response that needs to be triggered, okay? So phagocytes, remember, these are a special type of white blood cells. They digest and eat the foreign invaders, okay? Neutrophils are a type of phagocyte. So we learned about that in last lecture in our blood, that they're most abundant in our blood. They die fighting, so they're not meant to live really long, but they become phagocytic on exposure to infectious materials. That's why when we get infected or inflamed, they're the first ones to show up. Then we have macrophages, and then remember we have monocytes in our blood. So monocytes are made from macrophages. They've been activated. And free macrophages can go throughout the tissue spaces and invade, and then you have fixed macrophages. So we already learned about one of them, microglia. That sounds familiar from the brain. Microglia is a special type of fixed macrophage that only hangs out in the brain to help its immune system. And how does phagocytosis occur? So phagocytosis first, it fuses to a receptor and realizes that the passenger debris is foreign, and it kind of engulfs it, adheres to it. It makes these little pseudopods, these little looking like fingers that bring it in, forms a vesicle. In that vesicle, you have what's called a lysosome, sorry if you remember back from cellular biology. The phagosome then fuses with the lysosome, which, remember, the lysosome has a bunch of enzymes that are meant to break down what we don't want growing. Okay, so that lysosomal enzymes join up here and release their enzymes and break it on down to Funky Town. And then sometimes if it's something that we don't need, it like literally spits it out at the end, like that, as exocytosis. Another thing that is really important to know that neutrophils have this and phagocytes have it. It's called a respiratory burst. Its job is to kill things, basically. Okay, so some pathogens, when they're not killed by the lysozymes, they have something called the helper T cell that helps a helper T cell, and that's a different type of blood cell. It triggers the macrophage to be like, that didn't work. Let's try that again. And what ends up happening, it releases some cell-killing free radicals. So it actually does cause free radicals. You get something called hydrogen peroxide, which is the same stuff that we put on our cuts to make it stop bleeding and stop the infection. Also can increase the pH and the osmolarity of the phagolysosome, making it more able to be broken down. And then we have defensins, which come from the neutrophils, and they help pierce the membrane of the pathogens. So if a bacteria is coming, it really goes, the neutrophil goes, doo, doo, and puts defensin bullets into that wall of the pathogen, and then hopefully it leaks out, and the macrophage can come in and all chomp, 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 and get it on out, okay? They have a natural killer cells. So I said these are what I call, and I don't mean to be rude, but they're called suicide cells in most biology classes. So they're not phagocytic, but their job is to kill things, okay? So they can kill cancer cells. They've actually been doing lots of research about using natural killer cells to target cancer and virus-infected cells to stop it, okay? They can't, so the only problem with these is that they can't, the attack cells lack the cell surface receptors, so it doesn't really discriminate. And what it does, it induces something called apoptosis. And apoptosis is a programmed cell death. And it does that in virus-infected and cancer cells. And what it also does is it creates lots of chemicals that enhance the inflammatory response, so our adaptive immunity can come in and play a role. Okay, so inflammation. You got a paper cut, ow, worst thing ever in the world, right? What happens? You get red, you get some heat, you get some swelling, you get some pain. And sometimes that paper cut, I guess, is really super horrible, but in other ways it can be. You lose your function. So the red is the redness is because all these cardinal signs, the redness, the heat, the swelling, the pain, is because of different chemical messengers called cytokines that cause it. Okay, so we have stages of inflammation. So body's inflamed, something has triggered it. We have what's called the inflammatory chemical release. Then we have some vasodilation, which means the vessels dilate and increase their vascular permeability, which means they allow more things in. And those phagocytes, those Pac-Man that come and eat things out, mobilize and go do and come out. Okay, so inflammation, it's all about the chemicals, man. Okay, so chemicals that are released into the ECF is extracellular fluid. So outside the cell, these chemicals are released. So an example is histamine, so allergic response. Allergic response is inflammatory. So when we take things like Benadryl, we're actually trying to stop that histamine release. But the histamine release is a chemical mediator. And it's something that's exposed because the injured tissues or cells are coming in. Then we have kinins, prostaglandins, and cytokines, or prostacyclins. And these are different types of chemicals that help this process out. We also have a system called the complement system, which is like a paintball target, and we'll get that soon. A paintball targeting process that literally comes in. It sees that something's inflammatory response as started, and it causes things called complement proteins to go psh, 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 and start firing in a cascade. There's 3 different ways to have it happen, but there's a cascade that occurs, And from that cascade depends on what happens in the end. Sometimes they get tagged, so other immune cells can come and get it. Sometimes it just bursts the entire cell together that was attacked, and sometimes it causes an even more robust reaction. Okay, so what do these kinins, these cytokines, these chemical messengers do? They cause the vasodilation of the small little vessels, the arterioles in the area. Capillaries, they make them a little leaky, so there's more permeability, and they attract many phagocytes to the area. So vasodilation. Vasodilation is when I get that paper cut and it gets red. That's called hyperemia. The blood is being the area is getting congested with blood because vasodilation is happening. So if you think about it, if I put myself on a hose, what's going to happen? Not much, but if I start letting it out, more fluid can get to that hose. Okay, what does this do? More fluid gets around, so that we're bringing more blood to the area, more ability to push pressure on the walls. And what ends up happening is that some of that pressure causes fluid to leak out, and that's called exudate. That's that swelling. So sometimes when you get a cut, your finger will swell. Okay, and then swelling, when there's lots of water and excess fluid pushing around things, that could push on nerves around that area that got hurt, and you get pain. Okay, so this is a good thing. We need this to happen. It drives me nuts whenever it's like, bring down the inflammation, man. Well, yes, certain issues we can, but for a basic paper cut, just let it go. Your body's doing exactly what it needs to do. So vasodilation, increased permeability can occur. So pain can also result. So pain is one of the cardinal things of inflammation. It can also happen when toxins are getting released from bacteria or those chemical messengers, those kinins or prostaglandins. But there's some benefits of swelling. There really is, I swear. It's not just an ugly looking thing. So benefits of swelling include that it helps bring the foreign material out and into lymphatics so it can get filtered out. It also brings those clotting proteins and the complement system that I just talked about to the area to help isolate it from getting injured further or to prevent invaders so they can't spread out, so any foreign pathogens. Okay, and then we have phagocyte mobilization, which means that neutrophils come to the area first. It goes starts sending out all these chemicals. Macrophages come to the rescue. Okay, macrophages, remember, monocytes that have been activated become macrophages. Okay, so if it's inflammation is due to pathogens, then the complement system is activated. And then once the complement system is activated, what that does is in certain parts of the complement system, we have something called opsonization. And what that does is opsonization. It takes literally like a paintball gun. It goes, choo, 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 and it tags the thing that's been foreign invaded. And what that does, it tags it and has those antibodies that hook on and see it and destroy it or break it down or bring it to other cells. It's like a paint tag, like, oh, that person's right there. And then the antibody opsonization can hook on to the infected cell. Okay. So how do we get these phagocytes to mobilize? First, we have leukocytosis, which neutrophils come out. The neutrophils start secreting a bunch of chemicals that you're not responsible for knowing. We have marginization, which means that all these neutrophils start kind of conglomerating around the area. We have diapedesis. So all phagocytes have this ability to kind of like if you ever seen The Blob or if you ever seen Ghostbusters 2, it just kind of moves out. Diapedesis means feet. So it brings feet-like projections. And what it does, it squeezes in between those cells and helps get into the little spaces between your cells to bring things in and to chomp them out. Then we have the last part, which is chemotaxis, which is once this phagocytes are here, they're sending off this chemical messenger like, red alert, we're being invaded. Come on. So remember, as soon as this attack occurs, so first we got the neutrophils, phagocyte mobilization. Neutrophils are first to occur. Then the monocytes with their macrophages come in. Okay, and also they do some cleanup. So try to get things out. So here's a great example. This poor person stepped or somehow took a nail into their skin. Leukocytosis occurred. So in the blood vessels, these leukocytes, mainly the neutrophils, are responding. We get marginization. It hangs out. It starts lining up and getting ready to the area that it needs to go. Diapedesis. It grows little legs and goes through these little slits between the capillaries and goes shoom, shoom, shoom. Chemical messengers are singled out and says, hey, this nail is not supposed to be here. Let's cause some inflammation. Get the crap out of there. Okay. Same thing here. This is a great little thing here. This poor person, I know, so many accidents occur in this lecture. This person has had broken glass from a beaker because they didn't practice good lab safety. And then we got all the different cytokines that are coming out, okay? Cytokines, remember, are the chemical messengers that are responsible for telling the body there's inflammation. And what happens when there's an infection? Inflammation is different than infection. Inflammation is just the exudate. It's the swelling, it's the redness, it's the warmth. But when we know something's infected is when we get pus, which is gross. Pus is this creamy yellow mixture of dead neutrophils, because remember, neutrophils, their job is done. They phagocytize, they send other chemical mixtures, and they die off. So it's a mixture of the tissues that are in there, the dead or living pathogens that are still in there, and then the dead neutrophils. So it's just a conglomerate yuckiness. And what ends up happening is sometimes you get an abscess. So if anyone's ever seen an abscess, it's normally like this wall or this little cup that's made of fibrous tissue that gets laid down. It walls off the pus so it can't go anywhere. But imagine all that pressure going up, you need to get it surgically drained. And there's certain bacteria like tuberculosis, which is a lung disease, that don't get digested by macrophages, and they form things called granulomas, which is basically a fancy term for saying a bunch of macrophages that can't escape, okay? And then we got our proteins. We got antimicrobial proteins. These help. So remember, we're born with antimicrobial proteins. We don't need to learn how to use them. We're born with this baby. It's all natural, L'Oreal, Clareol, whatever hairdressing thing you want to say. Okay, so interferons are a type that we need to know about, and they kill viruses. And then we have the complement proteins. Remember, complement proteins activate. When they get activated, they enhance inflammation and destroy bacteria. They use opsonization. So opsonization is that paintball. They're sugar-coating the bacteria, saying, hey, we're right here. Come get it. It's right here. And the antibodies then see it, and they can attach the opsonization and bring it on down to Funky Town. Okay? Ooh, okay. Let's talk about a fever. Getting hot in here, so don't take off any of your clothes during this lecture. Okay, anywho's. Yeah, it's been a long day with patients. I'm getting goofy, guys. All right, so a fever is 100, anything above 100.4. And why is your body's, so remember, homeostasis. When a foreign invader's there, most foreign invaders, why does our body reach to 100.4? Because guess what? Most microbes can't survive past 100.4. That's why our body has this innate inborn ability to sense this and raise our body temperature. We can kill it and get it out, okay? So in my opinion, that fever is a good thing. I mean, there's a point where we say fever is a bad thing, but for the most part, we don't treat the fever, we treat the person who's having symptoms from the fever. The fever is a good thing. It's getting rid of the yuck. Okay, so now we talked about what we're born with. Now we're gonna talk about what we're not born with, and that's adaptive defenses. So adaptive immune system is something that we need to either learn or acquire. And what it does is it helps either amplify the inflammatory response or activate complement, okay? And it's shortcoming, which means that we need to be exposed to something for us to learn how to adapt against it, right? So need to know. Big, bold letters here. I know I'm doing like weird washy things. Okay. These are the characteristics of adaptability. It needs to be specific, which means it has to recognize and target a specific antigen. Antigen is what the body is a foreign, it tells the body that this is foreign. It has to be systemic. It cannot be restricted to the site of infection. It has to be going all around the body. It has memory, which means that once it happens, if it happens again, your body remembers it and you bring out antibodies or cellular immunity. And this is why we do immunizations. Not to get on the soapbox, but hashtag immunize your kids. This is what prevents measles, mumps, rubella. This is what prevents pertussis and tetanus. It is this triggering the body to think that it's had an infection so we can make antibodies so we don't get the infection, okay? And there's two branches of adaptive system. There's humoral, which is all about antibodies, and antibodies are proteins that help bind things and bring them on out. And then there's cellular immunity, okay? And it's not very funny to have antibodies. Ooh, okay. So humoral immunity means you're using antibodies, and these are made from those lymphocytes, those little purple, blue, black dots that you see everywhere. And they're circulating all over the body in body fluids, okay? They bind temporarily to the target cell. So something gets infected, they're gonna bind and they're gonna temporarily inactivate. They're gonna mark that So what these antibodies do is that opsonization, as I was telling you, is that they temporarily activate and they mark it for other cells to be like, hey guys, right here, bring it on out, Funkatown. Okay. Then we have humoral immunity, which also has extracellular targets as well. Okay. So extracellular targets are things that are outside of the cells. Okay. Then we have cellular immunity, which are the lymphocytes. Those are the things that we hear most commonly about. Okay. These are specialized white blood cells that can act against a target cell. So they can either act by directly killing the infected cell, or they can enhance all the other chemical messengers to get that thing going out. Okay, and then cellular immunity has cellular targets. We're going to remember that. Okay, we talked a lot about antigens. I think I've already told you what it is, but I'm going to tell you again. So antigens, they're a substance that the adaptive defense system doesn't recognize as self, and it targets an immune response. Okay, it could be a complete antigen or something called a hapten, which is a half antigen. They contain something called antigenic determinants, which are things that make them different from the rest of the body. It can be a self antigen. So self antigen is the whole thing with autoimmune disorders. The body can't recognize self versus different, so it starts attacking, like the MS that we talked about. It attacks the own body. It has something called immunogenicity, which means the ability to stimulate specific lymphocytes. It has to have that ability. And then reactivity, the ability to react with activated lymphocytes and antibodies released by immunogenic reactions. So we're looking at all different types of things that are coming from foreign invaders. Okay. So in the center here is your antigen. Out here's your antibodies. So antigen links to antibodies, and then it gets tagged for whatever it needs to be doing. Okay. So what determines an antigenic determinant? It's either the antigen, the binding site, or the antibody itself. Okay. As I said, in the previous lecture, I mentioned something about MHC proteins. These are on human cells. This is what recognizes our cells as human and belonging to us rather than anyone else to the outside world. Okay. It's called a major, MHC protein is a major histocompatibility complex. It's unique to the individual. So my MHC is different from your MHC. So that's why organ transplant don't always go by as planned, because maybe we have different MHC. We do have different MHC components. And what they can do is they have a little groove in them that can hold a piece of self antigen or a foreign antigen to be like flag, like, hey guys, I'm a part of Dr. Wood, or hey guys, that's part of Billy Bob. Okay. And then T lymphocytes are what's used here, and T cells can recognize only the antigens that are presented on these little proteins, the MHC component. Okay. So lymphocytes and antigen-presenting cells. So we have an antigen. We need something that's going to be like ta-da-da-da-da. Here it is. So part of adaptive immunity, we have 3 types of crucial cells. We have lymphocytes. They come in 2 flavors, B cells and T cells. Remember, B cells make your antibodies. T cells work with cells. And then we have antigen-presenting cells. So they don't have any specific antigens, but they play a role to help out with other immunity. Okay. How do these cells know what's self versus self? I'm so confused, right? So T cells go to school, and they go to school in your thymus, which is a little gland in here that's part of your immune system. We're born with it. As we get older, we don't use it anymore, so it becomes a fat pad above your heart. But the thymus, its job is to educate T cells to learn self from non-self. So the current working theory is that for autoimmune disorders, something went wrong at school and someone didn't pass their test, and that's why we can't recognize it. We have antigen-presenting cells. So antigen-presenting cells literally will eat up. So a macrophage will eat something up and then present its outside. It's kind of like a baby bird feeling. It's going to like eat all the food and then bleh, now see what I ate. It's on top of me. It's on top of me. Okay. So we have dendritic cells. We have macrophages and B cells. Okay. These are all antigen-presenting cells. So they bring the digestive stuff in. They put their antigens on top and flag for the other immune systems. So basically regurgitating what they ate and saying, I didn't like this meal, guys. Come take it away. Okay. Humoral immune response, antibodies. Okay. Antibodies, what do? They bind to antigens and they cause other cells to react. How do we figure out how to get antigen for the right thing? So we have an antigen. Antigen binds to the antigen-presenting cell. it causes a primary response. The primary response then causes an activated B cell to become activated. This becomes into a plasma cell, and it clones itself by making all the different antibodies when it first had a primary response. So example, you've never had tetanus before, but you probably got your tetanus vaccine. So what the shot did is it gave you an initial response by saying, hey, here's the foreign issue with what's tetanus is associated with. Then it's gonna, your body is like, okay, we recognize it. Let's clone a couple of things. Let's get some antibodies moving. And then if you ever get tetanus again, really tetanus, the whole idea is that those antibodies are ready to tag that antigen from the tetanus and stop it in its tracks. Okay, and as I said, clone cells. So what they're doing is not only they're making antibodies, but they're also if they're not making antibodies from plasma cells, what they do is become memory cells, and this is how we remember how to respond to antigens in the future. Immunizations, hashtag get them all. Okay, antibodies, there's different classes, and they respond to different things. So the major classes is what I want you guys to remember. So we have something called IgM, and IgM is anytime we have a reaction to something, IgM is going to be the first responder. They're going to be the first and most abundant apparent. So example, with COVID, if someone got infected with COVID, we'll be testing their IgM because that's the first one to respond for antibodies. What do? They activate complement, they opsonize, they get other people to help. There's IgA, which is only found in mucous membranes or other secretions. So IgA is found in breast milk and is found in the gut. We have IgD, which we don't really use as much. It functions as a B cell receptor. We have IgG. IgG, immunoglobulin G. So once that IgM comes up, now we need something a little bit more, not permanent, but a little bit more long-term. Then IgG antibodies are being made. And that's starting from secondary and late primary responses. And these are the ones that, if there's antibodies present, they can cross the placental barrier, okay? Placenta is where the baby in a utero forms. And then we have IgE, and IgE is mainly, remember we talked about eosinophils? So guess what, same thing. IgE is an antibody response to allergies and parasitic infections. What they do is they cause these things called mast cells, which hold all this histamine, which is the stuff that causes an allergic reaction. And when the IgE binds to this mast cell, boom, histamine, and you're sneezing, coughing, watery eyes, the whole 9 yards, okay? So as I said, immunologic memory, primary immune response, something, we get triggered by something, about 10 days is when the peak level rises, so we start to see antibody IgM first. That's your primary response. Then the secondary immune response is that more antibodies are being recruited to make memory and immunological memory, so we have antibody IgG. This is a nice little showing. I definitely want you guys to concentrate on this. So we have humoral immunity. We got active and passive. Passive is what you didn't have to really work for to get. So an example is that, so breastfeeding, hashtag please breastfeed if you can. If not, no, I mean, fed is best. But breastfeeding sends that IgA and IgG to the baby. So it gives them some natural immunity going there. Then we also have artificially acquired. So sometimes an example is that Kawasaki's disease is a disease that we're not really sure what's caused, but it requires a gamma globulin injection to give us some immunity. So they give you a dose of this gamma globulin injection. Then we have active. And active, we can do to naturally acquire it. So if you ever had chickenpox, you probably won't get chickenpox again because your humoral immunity has remembered what that varicella, that chickenpox looks like in your body. And now has antibodies that if it even comes in close vicinity and hopefully makes enough of it, you're not going to get chickenpox again. And then we have artificially acquired, which is that vaccine component. Okay. As I said, antibodies, what do? They do many things. So antibodies, what they do is they bind to the antigen. What do they trigger? They can either neutralize an invader by making it not pathogenic anymore. They can cause a bunch of other different cells to agglutinate or bind to it. Like that would be how a ABO blood type group would happen. Or they can also, antibodies can bind to antigen and cause it to precipitate out, and sometimes even just pee or take it out, whichever way you want to look at it. And then we have also that complement system. And I know I went over quite a few times, so I want to make sure you guys understand. The complement system is an innate ability, which is using chemicals to be able to tag things for it to get out. And this part right here, you can see that complement does a couple of things. It can either be enhance the inflammation process by bringing more chemicals on board. It could also trigger these phagocytes to come and chomp, chomp, chomp and get rid of it, or it could just burst the cell altogether and say, screw you guys, I'm going home, and then burst it. Okay? I feel like we've talked a lot about parasitic worm invasions, and I hope it didn't freak you out at all. But parasitic worm invasions, they're known as schistosoma and ascaris, and these are definitely worms to be worried about in the GI tract. And same thing with IgE. Remember that IgE is important in allergic response. So antibodies, summary, very important. Look at this side. Antigen antibody complexes do not destroy antigens. They prepare them for destruction by innate defenses or another defense. Antibodies go over extracellular pathogens. They do not invade solid tissue unless lesion is present. Okay. And what they do is that they activate mechanisms that destroy a virus in certain instances. Then we have, so those are adaptive, that's our B cells. Now we have our cellular response, or our T cells. So T cells, what they do is they fight against things that are inside the cell. So antibody are things that have attacked outside the cell. These things are inside the cell, okay? Inside the cell are viruses, bacteria. They can go in the cell and change the DNA or change how things are happening. Cancerous cells always starts from the inside out, or abnormal cells, or foreign transplanted cells. So when we have things like organ transplants, sometimes the cellular immunity does not work in our favor, okay? And then some T cells directly kill cells, and others release other chemicals that regulate immune responses, okay? So T cells, pee the food, wash your hands. Great way to boost your immunity there, okay? We have different types. We have CD4 cells, and these CD4 cells become what's called T helper cells. Their job is to be like, hey guys, they're kind of like the guy who directs traffic to say where everyone's going. And some of these CD4 cells become regulatory cells, which help moderate the immune response to make it not so robust or more robust. And then we have CD8 cells, which are cytotoxic T cells, okay? They have the ability to, so if that virus is invading a cell, it will destroy the cell and also become memory as well. Okay, so here's a look at all the different types of T cells. You can look at them in your own leisure. I am not going to quiz you on the exact chain of events here, but what I want you to know here is that first we have an immature lymphocyte. It gets educated at school. Remember, it goes to the thymus, gets its education about self versus non-self. Then it decides if it wants to become a CD4 or CD8. CD4, what it does, it helps flag things. So this antigen presenting cell here has an antigen. What happens is that it has a little hook here on the CD4 cells. Like, let me help you out. Let me take that off your hands. Let me help you. What does it do? It helps it out by binding. And by binding, what you're doing is you're getting a whole cascade of effector cells here. On the other hand, you have CD8 cells. They have an antigen presenting cells as well. And we use a little bit of the CD4 here to help, but mainly CD8. So it gets bound to the antigen. It's like, screw you guys, I'm killing y'all. Done. Okay. So this is what I kind of said in a nutshell. So remember, without T helper cells, there are no immune response. So we need those T helper cells to mitigate and help. Okay, so here's a great little thing that shows immunity. We have the T cells that bind to that antigen presenting cell. T cell then stimulates some other mechanisms to occur. And then the dendritic cell can either activate a CD8, which is a cytotoxic cell, or it can help out by getting more T helper cells on board. And I'm going to show you guys a video. Yes, it's from Storybots, but man, it's really good for simplifying what's going on here. My kid loves it. Okay. And then what are those effector T cells? They help amplify what our innate abilities to have our immune system do. And there's a whole bunch of different flavors. There's Th1 cells that are different subsets. There's Th2 cells. So Th1 is really cellular. Th2, what I want you to remember about that is they're a type of T cell, and they defend against parasitic worms and eosinophilic issues. And then there's these things called Th17, and those are actually really important for autoimmune because they're regulatory T cells. And then we have the cytotoxic T cells like we talked about. So these are found in the blood and in the lymph and lymphoid organs. So lymph nodes house these cytotoxic T cells, okay? That's just a little bit of them. Lymph nodes technically hold all the different types of T cells, but for this purposes, they infect the their targets are virus-infected cells, cells that have infected on the inside, not the outside, cancer cells and foreign cells, so transplants or transfusions. And I said these Treg cells, this Th17, Their job is to regulate the other T cells and other chemical messengers. Okay, when we talk about immune, we can't talk immune without talking about what happens when we don't have enough of this immune system response, or we don't have enough cells. So we have something called SCID, which is severe combined immunodeficiency, which means that your DNA does not code correctly for making B and T cells, and you have to have bone marrow transplants. Then we have acquired immunodeficiency syndrome, which is AIDS, and we should all be familiar with that, but if we're not, AIDS is the end product of HIV, which is a retrovirus that basically attacks T cells and makes it that you can't fight off infections in the end. Then we have autoimmune diseases. Autoimmune diseases for naturopathic medicine is our bane of our existence, at the bread and butter of our practice, okay? So autoimmune diseases that it cannot recognize self from self. So the body's attacking itself. Examples are rheumatoid arthritis, the body's attacking its own joints. Myasthenia gravis, we talked about that in musculoskeletal. It attacks against the acetylcholine receptors. Multiple sclerosis, it attacks against the myelin sheaths. Graves' disease is a type of thyroid issue that causes your thyroid to be overactive. It's not also mentioned here, but it's also Hashimoto's, which is the opposite of that, which is underactive thyroid because your body is fighting against your thyroid also. Type 1 diabetes is a type you're born with, and it actually attacks the body's own pancreatic cells. We have something called systemic lupus erythematosus that affects multiple organs because it affects the small blood vessels. And glomerulonephritis, also small blood vessels. Okay. And why does this occur? It's because of failure of self-tolerance. Something either happened in that T cell education school or something happened that caused our bodies to mimic something. So we don't recognize what's self versus self, okay? And then we also have things called hypersensitivities. So it's great to have the immune response, but it's also, that's normally harmless. But sometimes our immune system, even if it's not even autoimmune, sometimes the immune system just hyperactivates and can cause a lot more damage than good. And the reason this happens is because sometimes it depends on antibodies that cause immediate reactions, like the anaphylactic, like someone's allergic to peanuts. Peanuts are in the air, and they're like, can't breathe. That's because of hypersensitivity. Then there's also delayed hypersensitivity, which means that it's by T cells. So allergic response, this is one that you guys are going to have to be very familiar with because it can happen in your office. So what happens, something is an antigen that our body feels like is not happy with. Our body responds to it by making plasma cells. These plasma cells then they bind onto mast cells, and when that mast cell feels the antigen come back, boom, histamine exploding all over the place. Okay, then we have the different types of reactions. So right here, type 1, that's an immune response. That's hay fever, that's anaphylaxis. So it's mast cell activation. It's using a soluble antigen, which means something has to get through our walls into our blood vessels, and it's responding by histamine burst. We have type 2, which you see here. In type 1, we're using immunoglobulin E. In type 2, it's immunoglobulin G, but this is cell mediated, which means that the cellular response is being a little bit too activated. So whether it's platelets or complement or something else. So sometimes drug allergies like penicillins can occur this. Or we also have things, certain types of diseases I'm not going to make you guys responsible for knowing, but those are other types of things that affect for type 2. Type 3, the big difference here is that it's an antigen antibody so that it makes a complex that embeds into the vessels. This one is cellular. This one, type 3, yes, it uses immunoglobulin G, but it embeds itself. So we see that in serum sickness. We also see that in certain types of kidney diseases. And then we have contact dermatitis, the PPD thing that we needed all to get for school to prove that we don't have TB. We have poison ivy, chronic asthma, these type of things. These are type 4 hypersensitivities. They react with the T cells. They're either Th1, Th2, or cytotoxic. And depending on what it is, it activates the immune system and has a robust, more than needed reaction. Now that tuberculin test, the PPD, it's a local reaction, so we're not caring too much about that. Contact dermatitis, local. Chronic asthma, not local. Well, it's technically local, but deadly. Okay, so that's it for this lecture, guys. I'll see you on the flip side for lymphatics.